is going to be used, okay, in a biochemical analysis, where in biochemical analysis we want to separate and identify and purify proteins. Okay, we want them to be separated because in a sample, let's say we get a certain amount of protein, we want to separate them and compare. Okay, you can use this technique called electrophoresis. It is you are going to place the sample, okay, in between of two electrodes. In simple terms, you are going to actually place them between two electrodes. So let's say this is positive, and then this is going to be negative, and then they are connected to power supply. Okay, so you have a sample over there. Okay, so this sample, let's say I just represent this using a line. Okay, so if you have that line, okay, let's say you have a sample. So the sample, if you put in the middle, okay, depending on the charge, okay, and the size, okay, they are going to move. Let's say the sample has negative charge. They are going to move to the left. Okay, they will move to the positive. Okay, if it is negative charge. Then if they have positive charge, they will move to the right. Okay, so it depends on the charge and it depends on the size. Okay, so you are going to separate, yeah, the mixture, and then you are going to form, let's say, similar to chromatography. Okay, you can actually look at the separation, but do remember this method is used for protein, and protein, they are colorless. Yeah, you cannot actually just simply see them. So what we do, we need to add some chemicals so that you can see them under the presence of UV light. Okay, so the chemicals and so on in detail, you no need to find out in detail. Okay, but if you want to have a clearer picture of what is the thing that is being done, yeah, this is the clearer picture. Yeah, you are going to have support, and then you have the electrode and so on. You can read. Okay, but basically the sample is placed on absorbent paper or gel supported in solid base. Okay, a buffer solution helps to carry the ions along. The ions, what they mean is the amino acid or the protein. So the rate of movement depends on the size and charge. Okay, if you have bigger size, slow, yeah, it will move slowly. If you have more charge, of course, they are going to move faster. Yeah, so it depends. Okay, so larger ions move slowly, highly charged, they will move quickly. And you will get a series of lines. And do remember, this series of lines only appear when you apply chemicals. Okay, you cannot just simply see that like chromatography paper. Yeah, you cannot do that. Okay, and then for chromatography, we call it chromatogram. So this one, electrophoresis, we call it electro ferrogram. Okay, so it's going to be the electroferrogram might appear like this. Okay, and in order to see this, after you put the chemical, you need to see them under UV light, and you will see something like this, a brownish or yellowish color over here. And you can see from here, it's quite difficult to see, but okay, you can see that these two electroferrograms are similar with each other. We can confirm that these two are same. Yeah, why? Because you can see that this one appears here, and then there is going to be a little bit appearing here also. So this one can be used to identify proteins. Okay, to let's say compare. Okay, in one let's say one crime case situation or something, you want to identify proteins and so on. Okay, if you can actually find out this protein and this protein, yeah, and then you compare, you can actually get the evidence. Okay, and this one normally electrophoresis is done is considered to be a simple one. Yeah, because in crime cases they will not use this method. Yeah, this is like simple cases, like for example last time, okay, you can actually go and read about this. Okay, last time in Malaysia there was a case 2 years ago, or I think 2 years ago. There was a case where in Cadbury chocolate, because in Malaysia there are a lot of Islamic consumers, yeah, so Muslim consumers, so they don't eat pork. Okay, so one of the student researcher, yeah, who's studying in university, they were studying about electrophoresis. And in the university itself, okay, they decided to take certain food sample and try to run it over under electrophoresis and try to compare with a certain protein or something. Let's say, if you say this particular food is vegetarian, then there shouldn't be any kind of meat protein over there, yeah, or something like that. So they did this experiment and then they compared with series of food, and one of the food that they tested was Cadbury chocolate. And in one of their testing, they found out that Cadbury chocolate suddenly, 2 years ago, contains traces of pork DNA, okay, which was halal during that time. And it became a very big issue in Malaysia, okay, where how come pork traces can be found in a halal certified food, especially for Cadbury. It's a very big company, yeah. So it became a very big issue, and then they called out the sample, and then they checked, and then the Cadbury issued statement. Yeah, it was only that particular batch. They don't know what happened, and so on. So you see how electrophoresis, okay, can be even used, okay, to detect those samples by amateurs. Like normal students can actually detect all this. So this is some of the things, maybe if you want to recall, this one can be done or that. Yeah, can be done using a simple device. Yes, the device, okay, is quite difficult to set up and so on, but it's a mini device. Yeah, it's a mini device. I think if you had the opportunity last time, they used to have a demonstration or something, a school visitation by a group or something. They did show electrophoresis in wallet group. Yeah, so I do not know whether you managed to see that, but it's a small device. Okay, the device is like twice the or maybe is as big as smaller than iPad. Yeah, the device. The entire setup is smaller than iPad, and you can actually get it done, and it's quite easy to understand. Yeah, so this is going to be something you want to actually take note. So basically, if you look at electrophoresis, we have learned that amino acid, they are generally they exist as zwitter ion. Yeah, so if you have lysine, glycine, and glutamic acid, if you have over here. So first of all, let's actually look at the structure of amino acid first. Yeah. So here there is H over here. So it's okay, correct. But this one, there is a mistake. There is no H. There's a mistake over here. There is no H. Yeah. So this one I will add. Okay. But if you look at the general amino acid, they must have the carboxylic acid group. They must have the ammonia group. Amine group, sorry. So they must have carboxylic acid group, amine group, carboxylic acid group, amine group. Okay, so they must have hydrogen. The rest of it is going to be the R group. Okay, and do remember the carboxylic acid. Okay, the H plus will go here. Okay, to here to give you the zwitter ion. So the same, it will go here. All these are going to be zwitter ions. Okay. So negative and positive, they will cancel, so no problem. And I mentioned to you that whatever that comes into the R group will determine the acidity. Okay, not only acidity, it will also determine the polarity. So if you look at this R group, if let's say the R group is alkyl or H, okay, in this case, glycine is the simplest amino acid. So you see, this is going to be neutral. This is based on what we have learned. Yeah. So if you have NH2 group over here, okay, in lysine, then they are going to become basic. Okay, they are going to become basic. And if you have carboxylic acid group, then it's going to be acidic. Okay, so if let's say, for example, okay, from where you get the O minus and NH3. So originally they used to have carboxylic acid group. Okay, originally they used to have NH2 group. Okay, so when you mix with water, yeah, when you add water, okay, water will generally give you H plus and OH minus. Okay, so the H plus and OH minus, if you put glycine over there, nothing will happen because it's neutral. Yeah, so nothing will happen, so it will exist like that. But if you have water with lysine, okay, lysine is going to be base. Okay, so they will have the proton. Okay, they will accept the proton. So this H plus will come over here, and they will start to give you NH3 plus. So they are going to give you the positive charge because of that. And then from where, okay, you have this negative charge, okay, if you have OH minus, this H will go over there to give you water. So when the H went over there, okay, you are going to get O minus over there, which is going to give you the negative charge. So this is going to be positive charge into overall. This is zero charge, negative charge, okay? So now if I put them over here, let's say I put them in the middle, okay, and then I put positive and negative. Let's see which one is going to be represented by the electroferrogram. So if you look at that, okay, B will stay in the middle, okay. This is positive, so positive will attract negative, so C will go there, okay, C will go there. And then negative will attract positive, so lysine will come over here. So that is how generally it works. Okay, but the problem over here is, okay, you can see amino acid, okay, different groups, okay, they will go up, they will go down. So if you want to compare like this, is going to be difficult. We want them to move in one direction. So how do you make sure that they move in one direction? Okay, you can control. Yeah, you can control. Okay, by if you want to separate the mixture of protein, you treat them with certain chemicals to make them, all of them, to be negatively charged. You don't need to know in detail about it, okay, but basically you are going to change all of them into negative charge. So once you change them into negative charge, okay, all the protein, okay, will have overall negative charge. They will move to the same direction, which is towards the positive electrode. Okay, and the only thing that will actually determine the separation over here, I think, still size and the charge, but not the positive or negative. The how big is the charge? Okay, that is going to determine. Yeah. So in this case, larger protein will move slowly. Okay. If you have more charge, then they will move faster. Yeah. So the dye is the one that I said. You add the chemical so that you can see. But even though you add chemical, you cannot see with your naked eye. So you need to still use the UV light to see. Yeah. So this one, okay, you can go through this question. Okay, the answer is all given, but I have discussed this with you just now. Okay, this is the part under electrophoresis. Okay, so I have discussed about electrophoresis. Which part is important for electrophoresis? Okay, understanding the concept. Okay, that electrophoresis can be used to separate protein. Okay, it can be used to separate amino acid based on charge and size. That is in summary. Okay, that is in summary. So that is about